Hi students, if you like my channel, please press like button and subscribe my channels. Today we will discuss about the plasma membrane of the cell. Plasma membrane is also known as cell membrane. Where do we find the plasma membrane? It lies beneath the cell wall. Beneath the cell wall is very thin, less than 10 nanometer, and this plasma membrane, this layer contains the cytoplasmic content called cytosol. Okay, cytosol. The plasmic content, cytoplasmic contents is known as cytosol. Clear? Okay. So what is the nature of the plasma membrane compared to the cell wall? So cell wall allow all type of substances into the cell, but the plasma membrane it selectively allow the substance, thereby it is known as an semi-permeable membrane, selectively permeable membrane, since it allows only the selected substance needed for the cell. Okay. So at the event of that microscope discovery, and especially electron microscope discovery, we are able to study, the scientists are able to study the nature of plasma membrane. I mean the structure, chemical structure of the plasma membrane. They have studied in the red blood cell obviously, and they have come to the conclusion that the plasma membrane is made up of bi-lipid layer. The plasma membrane is made up of bi-lipid layer. Okay, two layers of the lipids. Clear? So it is having that head regions as well as tail regions. In case of head regions, and they are hydrophilic nature, and they are facing outside the cell membrane. Okay, and the tail regions are hydrophobic in natures, non-polar in natures. They are facing inside of the cytoplasm. Clear? So this is the chemical studies of the plasma membrane. So plasma membrane is made up of bi-lipids layer, that polar region, non-polar region. Polar region of the bi-lipid layer face outside of the cell membrane, and non-polar regions, tail regions, face inside of the cell membrane. Why the non-polar tail region face inside of the cell membrane? There will be reason behind it. Okay, because in order to protect the saturated hydrocarbons, the non-polar regions tail are facing inside the cell membrane. I means towards the cell content. Am I right? So that is the reason. This is the way that non-polar tail regions are protected from aqueous solutions, because this aqueous solution can degrade that saturated hydrocarbons. So thereby they are away from that aqueous solution. The hydrophilic contents are trapped on the head regions of the bilipids layer. Okay, so slowly the biochemical studies shows that this, along with the bi-lipid layer, there are certain substances found, that's an protein as well as carbohydrate. Along with the lipid layer, then protein and carbohydrate also identified. So the protein and lipids percentage may differ from the cell to cell. Okay, so examples, the cell membrane of the erythrocytes will have the 52% of that protein and 40% of lipid molecules. Okay, we are able to identify two types of proteins in the plasma membrane. One is embedded inside the plasma membrane, another one is outside the plasma membrane. So embedded protein is known as the integral protein, and which is facing on the surface of the plasma membrane is called the peripheral proteins. Okay, we are able to identify one more protein called channel protein, and which allows that protein molecules are the hydrophilic contents into the cell membrane with the help of the carrier protein. That's with the help of the carrier protein from higher concentration to lower concentration, the substances are entering from the outside of the cell into the inside of the cell with the help of the protein molecules, the carrier protein, known as a facilitated diffusion. Because the diffusion is nothing but the movement of the substances from the higher concentration to the lower concentration passively without any energy expenditures. So that's we call it a diffusion. So here we are able to see the different molecules. Okay, so when we see this diagram, this is the best model described about that Singer and Nichols in 1972. This is called the fluid mosaic model. So according to the fluid mosaic model, we have seen the mosaic floor. When we see the mosaic floor, we are able to find small bubbles, color bubbles, and different size and shapes, and different components are identified in the mosaic floor. The same way in case of plasma membrane, according to fluid mosaic model, we are able to find out the carbohydrate, we are able to find out the glycoprotein, glycolipids, along with that integral proteins and cholesterol. All these components are embedded. So thereby, this is a best model for the explanation of the cell membrane structure. Okay, so we are able to see that this is, we call it in quasi fluid structure. Quasi fluid. So what is the quasi fluid? The proteins are able to move overall with the bi-lipids layer in lateral direction. Lateral movement of the protein overall in the bi-lipids layer. This movement is known as the ability of the cell membrane to move the substances. This ability is known as fluidity. Okay, fine, that's good. So this is measured as a fluidity, the ability of all the lipid molecules are able to enter into the cell membrane from the outside to inside, and it's able to move from the vertical directions. So that this movement is known as a flip-flap movement. Flip-flap movement. So flip-flap movements, all the lipid membrane, lipid molecules are able to move easily inside the cell membrane. However, and certain hydrophilic components are not able to move into the cell membrane, and proteins are not able to move into the cell membrane, and they are able to be facilitated. Okay, so the smaller substances are able to move inside the cell membrane. The larger substances are not able to move inside the cell membrane. They need special movements as well as the facilitated diffusion, osmosis, as well as active transport. So all these movements help the protein molecules penetrate into the cell membrane easily with the channels, with the help of channel proteins. Okay, fine. So what is the function of the cell membrane? The function of the cell membrane is, one is in transport of the substances, another one is in signal, cell signaling, cell transductions. Okay, how these cell membranes helps to transport different types of the substances into the cytocells. Okay, fine. So let's discuss about one by one. One is in diffusion process. Okay, what is diffusion? The movement of the substances from higher concentration to lower concentration. They do not require any energies. Okay, so they are moving based on the concentration gradient. Always the concentration gradient maintained in the cytoplasms inside the cell. That's why the substances are able to move into the cytoplasm of the cell. Okay, from higher concentration to lower concentration. So this is, we call it a passive method of the transport. Okay, so certain cases and certain bigger molecules are not able to move, and they are able to move into the cell membrane with the help of the special protein. That's we call it a carrier protein. So these carrier proteins, once it bound with the cell membrane, they make a channel. So these channels are allowing the different hydrophilic substances into the cell membrane. So thereby into the cell, that hydrophilic content are able to easily penetrate. Okay, fine, that's good. Then another one is an active transport. In case of active transport from the lower concentration to higher concentration, and it's not possible. Okay, it requires more energy. So thereby, with the help of the ATP, the substances move from the lower concentration to higher concentrations of the cells, is known as an active transport. Active transport require energies. So this is an ATP dependent, energy dependent process. Okay, fine, that's good. Okay, let's move on to the next one. Is an endocytosis exocytosis. What is an endocytosis exocytosis? Endocytosis, once the molecules bound with the cell membrane, the membrane fold and make a vesicle form. After the vesicle form, and it sends this vesicle into the cytoplasm as a vacuoles. So the vacuoles store these substances and degrade it for the purpose of the cell. Okay, so the vesicle formation, then vacuole formation, then the degradations. So endocytosis, the larger solid substances or liquid substances are able to enter into the cell membranes through the form of the vesicles or vacuoles. Because this membrane, once the larger substances bound with the membrane fold many times and form the vesicles. Okay, then allow the vesicles penetrate into the cytoplasm. In the cytoplasm, it develops into the vacuoles, and these vacuoles and store whatever necessary, okay, for the purpose of the cell activity. So exocytosis expel out, okay, the vacuoles attaches with the cell membrane, then the foldings happen, then finally it rupture outside of the cell membranes. Okay, so that is exocytosis. Exo means outside, okay, endo means inside the cytoplasm, outside the cytoplasm. Okay, fine. So this endocytosis is two types. One is an phagocytosis, phagocytosis. What is phagocytosis? In case of phagocytosis, the solid molecules bound with the membrane fold many times and form the vesicles. Okay, the vesicle enter into the cytoplasm. This is a phagocytosis. In case of this is phagocyte. In case of phagocytosis, solid substances are entering into the cell. But in case of pinocytosis, what happens? The liquid substances enters and form a vacuoles. So the liquid substances enter into the cell membranes, form as vacuoles and store. At certain cases, certain times, and it degrades and rupture out inside the cytoplasm. That is an pinocytosis. Okay, so next one is in cell transductions or cell signaling. So cell signaling in case of plant cells and fungi, and cell signaling is playing a major role. Once the substances bound with the cell membrane, the first messenger, which is having the receptor of the first messenger, receive the information and send to the second messenger, okay, in the cell. Okay, fine. So for that reason, the nitric oxide, okay, or nitrous oxide is playing the chemical messengers, and it is helping the signal to be transported from the first messenger to the second messenger in order to perform the function of the cell. So this is, we call it in cell signaling. Cell signaling we will discuss in the details in the later modules. Thank you.